Alrighty. Well, I want to welcome Kelsey. Thank you, everybody, for hanging around with the difficulties. I really, really appreciate it. So, we're here to talk to Kelsey Woodruff, and she is a. Well, first off, sorry, what are your pronouns?
She, her's fine.
She, her? Okay, I just wanted to make sure.
Yeah.
Um, so, Kelsey is a PhD student at the University of Washington, and you are a cell biologist?
Yeah, yeah.
Cool.
So I'm in molecular and cell biology.
Awesome. So tell us a little bit about your research. I want to know, like, before we get to talking about like what you study and how cancer changes and affects women's or not women's, sorry, people's bodies. I woke up late today. I meant to wake up at 5. I woke up at like 7:45. So I was like, sorry. I'm still waking up. Hence the
That's
coffee.
that's fair. I am also still waking up. I woke up about 7:40
Yeah.
today. So
Hence the coffee. But so what was your route to grad school? Like, what made you realize you wanted to be a cell biologist?
yeah, so I love getting this question because I get to tell my favorite joke, which is that I'm a reformed pre-med. So when I went to college, okay, so just backing up, I grew up in an area that didn't really have a lot of access to science, a lot of access to research. So I didn't really know that this career, like this grad school journey I'm on now, existed before I went to college. But I knew I really loved science and really loved cell biology. And I was like, doctors do science, so kind of. So I went to Ball State University in Indiana to study cell bio. Uh, and really loved it. Along kind of like my pre-med route, I was like, oh, I should start doing research, get some experience in the lab, learn to think in a different way. And was really fortunate to have a really fantastic mentor through that experience. And, I think it was about my junior year of college. I was studying for the MCAT, and nothing will make you consider what you really want to do in life like studying for the MCAT.
I believe that. I absolutely believe that. I once too wanted to be a medical doctor. Nope.
Yeah, yeah. So, um, had to sit down. Like, I was waking up every day at like 6 to study and then go do a full day of classes. And I was like, I don't even think I want to be a doctor. So pursued different research opportunities. Like, I did like a summer internship, and then the fall of my senior year, I applied to grad school and moved to Washington, and that's where I've been ever since. So
So, are you from Indiana originally?
yeah, yeah, I am. So I grew up and
Like,
went to college in the same like 40-mile radius. So
uh,
Washington was a big change.
were you excited about that?
I was so excited. I had wanted to move to the West Coast since I was like a little kid. So it felt like a big like goal fulfillment
Yeah,
in a lot of ways to be able to move. So
I know what you mean. I know what you
yeah.
mean. So when you went to grad school, and this is something I know, like I talked about being interested in understanding, because grad school for biology is way different from grad school for
Yeah.
geology. Like I know
Yeah.
you and Balint, you're both biologists, and so Balint is science streams. If you guys aren't familiar, ooh wait, I have to tell him I'm okay. But how? So in geology, we don't have like lab rotations or anything. You kind of just like, you know what? I want to study earthquakes, so I'm going to go work with a seismologist, and then you
Mhm.
get accepted and you go work with one person. How does it work in biology? Because there's like different rotations and stuff. It sounds really complicated.
Yeah, yeah. I think it really depends on the program that you're in, because I have heard of programs like that where you're directly admitted into somebody's lab and then you work on one project for your entire PhD and graduate and you're an expert in that field. The majority of the programs that I applied to, because I went to grad school straight out of my undergrad studies, and I did my undergrad in a smaller research university, so there was definitely less exposure to the different areas of biology and different like kinds of research you could do. So I applied to programs that had the rotation model. It's basically like in your first year, you get to pick. anywhere from like 3 to 6, depending on the program, different labs and work there for like 5 to 10 weeks and see if it's a good fit, see if you like the research, see if you like your mentor, see if you fit in well with the other members of the lab. And then you can make the commitment to um, actually joining and like pursuing your PhD.
I kind of like that because you get to bounce around and like I mean I've had a friend who had a very bad advisor and she ended up changing advisors. It was a hullabaloo. I kind of like this
Yeah,
where you go and see where you fit in best.
yeah.
What were the
Um,
different things that you like went through that you decided you didn't care for before you committed?
yeah, so before I even started grad school, so I moved to Seattle the summer before grad school, and I had like a temporary tech job, which I was like very fortunate to have, and that was working in genetics and evolution. And I was like, oh boy, I don't think that this is something that would be a good fit for me long term. So and it was like nothing against like I really loved the lab, I really loved the environment, but it was just like this subject is just like a lot to wrap my head around. So kind of, it was a good opportunity to do that and see like, oh, maybe this isn't it. I also did a rotation on a more like tech development, so like drug development side of things. And that it was cool work, but I was like, I don't feel like I'm getting to like stretch my scientific legs as much as I would like, and getting to be as creative as I could in other ones.
Yeah, I feel that. I'm working in a not research position right now as my daytime job, and
Yeah,
science is much more preferable. It's a lot more
yeah,
fun. It's a lot more
yeah, for
fun.
sure.
Um,
Um, I got a message from Kirsten at Fred Hutch. Can everybody hear me okay? Like, are my headphones coming
they're going in and out. I've turned the
through? Okay.
audio up, but it just does go in and out a little bit.
Okay. Maybe I can just switch to using my computer microphone. Sorry if you hear my cat because of this.
We love that. Bring the cats in. Bring the cats in. We have a strong cat community. We
Yeah.
even have a. Oh, that's worse. No, no, no.
That's
Headphones back.
worse.
Headphones back.
Okay. Okay.
I did that my last stream. There was some of that with the reverb, and I went and bought a cheapy pair of headphones because I couldn't get my like Bluetooth ones to connect with my computer.
Oh, and all right. I need to
I think
repeat
that it's still
that.
set on your computer microphone.
See.
Your cat's here helping right now, Kirsten.
Is that
we
better?
had a we had a beginning of stream appearance. I cannot hear you
Is that
now.
better?
Much better. Great.
Oh no.
Thanks.
Okay, good.
Yeah, thanks.
Yep.
Um,
Sorry for that headphone detour.
no, no, it's fine. It's
Yeah.
fine. That's literally like part of live streaming. If it's not a
Yeah.
good stream if you don't have at least one technical difficulty. Like it's just not real. You're faking.
Fair.
So,
Good to know. This is my first like streaming experience. So this is
oh, good. I'm glad. Well, I hope you enjoy and having fun. We
yes.
have a couple questions in chat. I do want to make sure we get to, but it's kind of like later on. I do want to ask, what biology are you studying or researching on? Daniel Sargent would like to know, is it human or cells or what?
Okay, so both. So I am studying acute myeloid leukemia broadly. So that's a type of blood cancer. It's more common in adults and it's a very aggressive form of blood cancer. And something that's kind of unique about it is that it's really heterogeneous. So there are a lot of different things going wrong in your cells that are contributing to the cancer. So I'm trying to look at a cellular level what things are going wrong that maybe we haven't characterized before, and how can we target those in the future with drugs or things like that.
Okay, so you're
Yeah,
really just wanting to know like why do these things change? What is causing cell changes no matter
yeah,
what?
yeah, exactly,
Cool.
exactly. Yeah.
That's really, really cool. What
Yeah.
I know that for anybody in chat who might not be a traditional scientist, you and I are not white males. We are
That's true.
female
Yeah.
presenting, but nonetheless, science is predominantly male and upper class and wealthy and white. What was your experience going through grad school not being part of that population? Like, were you low income? Or do you identify as
Yeah.
anything that's not traditionally a scientist?
Yeah. So I definitely identify as somebody who comes from a low income background. Um, and it was definitely challenging. I think like it's gone away more in grad school. One of like the really nice parts of grad school is that you get paid for the things that you're doing, which has been just kind of huge for me. Whereas like, in undergrad, it's a lot of voluntary research experience, which like, you know, has a lot to do with the funding. Like I know the people I worked for, if they could have paid me, they would have a lot of the time. Um, but it was hard. It was hard to kind of find a niche to fit in. It was hard to find a mentor initially. Like I really struggled, I think, with my first year of college, just not feeling like I fit in with the other people around me. And then, you know, I feel a lot of like fondness for my first research experience because that professor was somebody I could really go to and be like, "Hey, like I don't know what a fellowship is. Can you like explain this to me? Can you like, I really like research, but I have no idea what like going to grad school means or would look like. Like none of my parents have ever gone to grad school. I don't know anybody with a PhD besides my professors. So
It can be
yeah,
really
it
it
was
can be really like scary, and you don't understand, you don't know how to do things. Like,
yeah.
what was it, just being able to ask these questions that made them a good mentor, or what was it really about them that you clicked with?
Yeah, I mean, I think like we had very similar backgrounds in a lot of ways. So I think, I would go through things that he had gone through, 20 years before, and he'd be like, "Oh yeah, that was really hard. Like here's how I got through it, and here's some things that like you can do as well." I think he was also just a very huge advocate for his students. So, he helped me apply for um, some like research funding as an undergraduate. Ended up being successful. So I did get paid a little bit of money, which was way better than getting paid no money.
That's awesome.
Yeah, yeah. And he was a very big advocate. He took the whole lab to like a state conference to present research and kind of connect with the broader community. So I think just things like that really helped me find my footing in science. And I don't think I would be in grad school right now had I not had those opportunities.
Sorry, I love that. I feel kind of the same way. I had a similar type of undergraduate experience with an amazing
Yeah,
mentor. So it's so,
yeah,
you really need that. And I one thing, and I don't know if you'll agree with me, and feel free to disagree. No is always a good answer
sure,
too.
yeah,
Um, but I feel like, it's less about when you've got students or people who want to be a scientist. It's not Things aren't so much about retention and getting people to want to be scientists. It's more about helping them to figure out how to do that and keeping them from being edged out, because it's really easy to be disenchanted and give up. And I feel like a good mentor, like you're an undergrad advisor, seemed like he was very supportive and wanted to make sure that everybody had those experiences.
yeah, yeah. I think like reflecting back, like I totally agree. And that like, it was never a question of like whether I wanted to be a scientist, but if you don't have access to, resources like if you don't have access to people to ask to be like, hey, like, how do I become a scientist? Like, what does this look like? What do you do for your job? Like, I think grad school and like research are really hard things to conceptualize if you've never met somebody who's engaged in those things. So yeah, completely agree with you there.
Yeah, for sure. So when you're in grad school now, and
Hmm.
you're a second year. Have
Hmm.
What does that look like for somebody who is new to the concept of grad school and how things work, and where in the process of getting your PhD are you? Where in the process of your research are you? And also, I consider people without PhDs to still be scientists. If you are practicing science and you're
Oh yeah.
working on it and you're actively doing it, you're a scientist.
Yeah.
Yeah, tell us.
Yeah, so I think I'm still pretty early stages. So the average time for a degree in my program is 5.5 years. So still on the front half of that 5.5 years. Um, yeah, I don't know. I mean, it's been challenging. I think like a lot of the beginning of a PhD is just trying to find something that will work to study. There's a lot of failure in science. Um, but I feel like I'm getting there, and I feel like my project is definitely on the right track so far. So
Cool. Um,
yeah,
I know I want to talk to you about your research because that's really in the nitty gritty of how cells and how cells change and how other things can affect cellular change and what triggers that and how cancer works and all that jazz. But before, I
yeah,
want to ask a question that Nice Opinion had. He says, or they say, I'm curious, what is the day to day like for people at research centers? Is that something that you
yeah.
can
But
answer?
yeah, I can totally answer that.
Cool.
Um, I can tell you what my day today is like. I mean, I think it's totally different depending on what your role
Oh,
is.
yeah.
If my PI's job is way different than my
Yeah.
job. Um, So I am strictly on the research side of things. So I don't have any patient contact at all. So I normally get to lab at like 8:30 in the morning. I try to start by like catching up, answering emails, organizing, planning, if I need a protocol, I'll print out my protocol. Uh, so that's usually like the first hour of my day, and then I start doing experiments. For me right now, I do a lot of cell culturing, so I spend a lot of time um, moving liquids into tubes and adding things to them, um, and then counting the cells to see how my treatments affected their growth or chemoresistance or whatever. Um, and that's the majority of my day. And then I head home. I try to head home at like 5 or 5:30. I'm not trying to be a scientist that's in the lab for 60 hours a week. I think that's
Good.
a great way
Good.
to burn out. Yeah.
Boundaries. Boundaries.
Yes, boundaries are so important.
Mm-hmm.
Um, but yeah, I think every day is a little different. I'm not doing the same type of experiments, which is a good thing because I think it means the research is moving forward a little bit. But yeah,
Cool. Thank you. Yeah, thanks for
yeah.
asking Nice Opinion. That's a great question. So tell us more about your research. What do you do?
Yeah.
What topic do you study? And then how, I mean, because I already know, but I know chat does not. And then could you explain how, that process works? And we have
Yeah,
another question. Do you make the experiments or does the team just kind of assign it? Ooh, that's a great question.
yeah, that is a great question. I think it's a little bit of both. So a lot of labs have certain techniques that they're really proficient in. So like for my lab, our major technique is flow cytometry, which is basically using lasers and fluorescent proteins to determine like what attributes a certain cell has. Um, it's kind of cool.
That's really
So
cool.
yeah, yeah. So we do a lot of that. We also do a lot of fluorescent microscopy, or we're, moving in that direction.
What is that?
Um,
Like fluorescent microscopy. So, chat, I
yeah,
can tell, microscopy, things under a microscope, but what makes it
yes.
fluorescent? What does that tell you?
Yeah, so we use either different kinds of dyes or different proteins that are tagged with things that will emit light when you hit them with a certain wavelength of laser. Um, So yeah, you can put that under a microscope and then you can, depending on what you've labeled in the cell, you can see where it's going in the cell, like if it's in the nucleus or the cytoplasm or on the outside of the cell. And you can kind of draw conclusions based on what you've labeled and whether or not two things are co-localizing or
That's pretty
um,
cool. That's
yeah,
very
yeah,
cool. How
yeah.
does the
Um,
laser does hitting the cell with a laser like move the dye inward? What is it? I want to know more. I know
oh my
it's
gosh.
such a just a
It,
weird random thing, but lasers are cool.
I was so bad at physics, so I don't know if I would be really
That's fair.
good at
That's fair.
articulating how it's working exactly. Um, so it doesn't move necessarily, but it like gets excited, and then when it gets excited, it like emits a certain
Ah,
wavelength of light,
okay,
and then you
okay,
can
I get
detect
it.
that. Yeah, yeah,
It increases the energy level, and then once it drops back down, it gives off the extra protons as a light.
yeah,
Gotcha.
exactly,
Cool.
exactly.
That's so
Um,
cool.
Yeah, yeah. So to get back to the question, so our lab has techniques like that. Other labs have other techniques that they do all the time. And then I think it's really up to a researcher and up to the project for how they're going to use those techniques to do things. So when I was starting out, it was a lot of like my boss would be like, "Oh, go do this experiment," and I would go do it. And then now that I have a little bit more experience and a little bit more training under the belt, I'm like, I think it would be interesting to use this tool to look at this thing. So I'm going to do this experiment. So it's more independent in that way. So
That's really cool. So they tell you kind of like where you're going, but you get to craft it. You get to design the experiment. You make the
yeah,
decisions.
yeah.
You and when you were talking about printing out a protocol, is that what this is in the morning? You come in and you figure out what you got to do that day, and that's the protocol you're printing out.
Yeah, yeah, pretty much.
Oh, that's really.
Yeah.
It's so different. It's so
Yeah,
different.
yeah, it's like I don't know, you could think of it like cooking and like I'm printing out the recipe for the thing I need
Oh,
to make in
okay.
the
That
morning.
does make a
Yeah.
lot of sense.
Yeah, yeah.
That does. So, what is your research about?
Yes. So I have a cartoon that I can send you. Again, maybe you can share your screen.
Yeah.
Would that be good? Okay. Um, I just sent it
In
over.
Discord?
Yes, in Discord.
Okay, cool. Open link. All right, let me do my OBS magic, hopefully. Browser? No. I'm just gonna do this. Uh, it's easier than trying to mess with OBS, honestly. Here we go. Great. So we have this cartoon. Awesome.
Okay,
Tell us
great.
all about it.
Um, Great. So I guess on the left side is what is supposed to be happening normally. So you can see this kind of like long blue thing with some hairs poking out. That's a cell surface protein called a heparin sulfate proteoglycan.
So this
And
thing
a
right
little,
here, the proper heparan sulfate. What does that mean?
yeah, I'll get there. So that whole thing is called a heparin sulfate proteoglycan. They're on most of your cell's surfaces, and they have the little things that look like hair up at the top are sugar chains, and the type of sugar is called heparin sulfate. And those sugar chains can be modified in certain ways. So they can be more or less sulfated, and they can also be sulfated in different locations. So those modifications allow it to interact with things on the outside of your cell. So that would be like what's shown in pink, the little ball right there, and you can see it's interacting
Uh huh.
with those chains. Um, I kind of like to think about it as like, outside of your cell is really, really chaotic. So you can think of a bunch of like tennis balls bouncing around a room. and then there's like a wall with Velcro on it. And some of the tennis balls are going to stick to the Velcro.
Oh,
And so the pink ball, those are called growth factors. Those would be like the tennis balls, and then the outside of your cell has these little Velcros, which I think of like heparin sulfate.
okay.
So yeah. Um, so depending on the modification, that changes which tennis ball the Velcro can interact with. It's an
Oh,
imperfect metaphor. Velcro is kind of one thing, but your cell surfaces are a lot of different things.
it works.
And then once it's stuck with the Velcro, it can go to the receptor, and then the receptor tells the cell to grow. It tells the cell to release certain signals, telling its environment to grow or the cells around it to grow. Um, so in that way, these heparin sulfate proteoglycans are really dictating um, the outcomes of your cells. So
Okay. So the receptor is what, like the information the receptor receives tells that cell and other cells around it what to do.
yeah, exactly,
Do each one of your
exactly.
cells have one of these, like, kind of little hand thingies that tells you what to do?
Yeah, each one of your cells has like hundreds of these different hand thingies telling it what to
Oh,
do. So it's really like information overload sometimes. And like as a person, it can be really hard to conceptualize, your cell is getting dozens of different signals all the time and having to decide which ones to prioritize, what to do, if it's coming from like one receptor versus another receptor. Um, so yeah, it's a lot of different things.
Ningao says only a dozen. Hey, dude, how are you? Welcome. And Daniel wants to know, is that how small deadly viruses like COVID-19 spread? It's a great question.
Yeah, so only a dozen, it's probably way more than a dozen. I'm just, being imprecise on the stream. I don't know the exact number.
That's fine.
Um, yeah. In terms of COVID, yeah, so COVID is, I think it does have a receptor mediated entry. And I'm not sure if it's COVID specifically, but these molecules that I'm studying do help some viruses enter into your cells. So they do mediate interactions with some viruses versus others and help them get into your cells. I don't work on that. I couldn't speak to it more in
That
detail than that, but yeah.
that's the mark of a good scientist.
Yeah.
Honestly, I don't know is the best thing that you can say when you don't know. That's what gets people trust in us, you know?
Yeah, yeah,
Cool. We have,
yeah.
as an undergrad in aerospace, I'm having trouble picking an avenue of research in propulsion. I see so many papers that indicate problems in some theories. How did you finalize picking your research topic? Great question, nice opinion. Were there
Yeah,
papers that kind of pushed back? Great question.
that's a good question. I think this might be something that's really different in the field. When I was picking a research topic, I knew I was interested in some aspect of cell signaling in cancer. Um, but I really, it could have gone 100 different ways based on those interests. Like there's just so much that's unknown about the biology. And so I really prioritized picking a lab to join based on how well I felt like I fit with the mentor and fit with the other people in the lab. Because, I'm probably not going to be working on this project forever. And I really want to make sure that I don't know. So it was like less important to me and for a long-term career to pick something that I'm gonna like stick with for the rest of my life, but it was really important to me to make sure I have a good experience
Oh,
in grad school and that I'm
absolutely.
supported. Yeah. Um,
If
as
you
far as
like something but you're not having a good support system, it doesn't matter.
yeah,
Yeah.
yeah, exactly. As far as like papers that push back on this idea, this is a pretty well-established model for the proteoglycans mediating these growth factor receptor interactions. Um, where I think I find the most pushback is that, I'm looking at specific modifications. So if I can send you another picture to kind of help me explain the modification aspect of it. And there's just not much published on how these modifications are are impacting things. So that's where I'm coming in and trying to like establish.
Oh, sorry. Thank you. I'm muted. Y'all,
Sorry.
chat. You are dropping the ball. Chat. Oh, they're in an ad break. That's why. Okay, so I've got this up. I've got
Okay.
this. That's not it. There we go. Okay.
Um, so yeah, so over here we're kind of like looking at the same thing that we were seeing before with the proteoglycan binding to the growth factor and bringing that to the receptor. But now we've zoomed in on these hair structures or hair looking things in the cartoon that are the sugar chains.
So these
And
little
you
hair
can,
thingies, that's
yes,
what these look like.
yeah,
Oh,
yeah.
cool.
So that's the chemistry behind it. And you can see circled in the red and the salmon and the pink are the different sites of modification.
Okay.
And so what I'm doing with my project is getting rid of, all of the types of modification that are in the red circle
Mhm.
and asking, can these cells still proliferate? Can these adhere to different proteins or different cells? And do these survive as well under stresses like chemo?
Oh, okay.
Yeah, yeah.
Cool. So you're actually are you are you putting the I actually don't really know much about how chemo drugs work. I know that they're awful and they wreck hell on your body.
They
Um
sure do.
what what does getting kind of into the cancer, and if this is too early for that, feel free to be like, well, we'll talk about that later. Um I, how do changes happen in these sites? Like, how do changes in these sites translate to cancer?
Yeah, so when I'm introducing the changes in the lab, I have the advantage of having a tool to be able to knock out the protein that catalyzes these changes. So I'm just baseline like deleting the thing that performs 2O sulfation and asking the questions. And that's like an advantage of the control of the research. We do know, like, the reason we're interested in this in the context of AML, is because we do know that some of those enzymes that are doing these modifications, so the the thing that's doing the modifications in the red circle or the pink circle, they're expressed at a higher level in AML, in some subtypes, and patients with those subtypes fare worse, so they have worse overall survival, um, and some more other like poorer outcomes compared to patients who don't overexpress them.
Thank you. I love
Yeah.
you. Thank you. Thank you. I love you all. I am on mute. I was like, oh my God, we have our first donation, $75. Holy smokes. That's
Oh, thank you.
amazing. Thank you so
Yeah.
much, Anonymous. Seriously, like that goes I know it doesn't seem like a lot, but like just $5 will buy Kelsey like 100 test tubes. My guest was just
Wait, I
sitting
think
there
I
like,
have the
uh, thanks, sushi. Um, And then let's see, $10 is 20 microscope slides because like grad students, we got to have funding.
Go through a lot of, yeah. Research is expensive. It's
Research is, and I couldn't, like, stuff for me is much less expensive, I'm sure, than stuff for bio. Like, you guys have
yeah.
to, I don't even know
I'm
how.
curious to hear about some of the stuff that you do. So you do field work.
I do some field work. I do some field
Okay.
work, and that usually involves going out and like collecting rocks and stuff. And then
Yeah.
I bring them back to the lab, crush them up, um, and then I can use different techniques like to measure their chemistry. We make thin sections, so put rocks on a slide and then look at them through the microscope. And that can tell us
Hmm.
some stuff. We do a lot of microprobe work to get mineral chemistries. And then from that, I can estimate, like using different voodoo, experimental voodoo, um, can recalculate the pressures and temperatures inside of a magma chamber that those
Oh,
minerals
wow.
crystallized at. And then you can look at things like gas contents and melts of little bubbles of liquid that were trapped in as the crystals were growing. Things like that can allow us to understand more about what actually triggers the volcanic eruption. So that's kind of what I do.
Oh,
I
interesting.
yeah, and one of the things I'm working on right now, or about to kind of restart, is a project on this really big eruption in Tonga last or in 2022. Gosh, it's like almost 2 years ago now. And it was like basically Krakatoa, but like yeah, I know you can look it up. Look up like Tonga eruption on YouTube, and it was captured on satellites, and amazing
Oh,
because this is the first. It's called a Phreato-Plinian eruption, so like super mega explosive water explosion eruption that like
oh.
don't happen. Maybe once a century or so, maybe. And it was the tallest eruption plume recorded to date. It went like 58 kilometers, like halfway to space.
Oh, that's crazy.
Yeah. So I've been working on that. And one of the things we're going to be doing is looking at the thermodynamics of that explosion because when magma and water interact, it's essentially a nuclear explosion. It's like the
Oh,
nuclear
wow.
chemical reaction.
Yeah.
Um, and like, yeah, I'm interested in using the energetics behind that explosion to try to figure out different. I'm being cagey on stream on purpose, you know. But no, it didn't go halfway to Uranus. It went halfway to space anus. Um, sorry, DL. It didn't go halfway to Uranus. Oh my God, sushi! Thank you so much for the donation. But yeah, that's a little bit about what I do. Looks like
Yeah, that's cool.
Guppy's wife is a geochemist and volcanologist. I would love to know who your wife is after stream, like in between us, because we might know each other. We might know each other. And there was another $20 donation. Oh my gosh. Thank you so much, Sushi. That $20 literally, that's like 40 microscope slides. And I mean, $150, that's groceries for an entire week because like with childhood cancer, like I mean, Kelsey, how specialized do the doctors have to be?
Yeah, so pretty specialized. So I don't know. I mean, I can't speak to the training just because I'm not, going through the medical residencies and fellowships. But Fred Hutch is a bone marrow transplant center. So when you talk about being able to buy groceries for a week for a family, the reason that that's important is because these families have to be quarantined basically because bone marrow transplant really compromises a patient's immune system. So they have to go to kind of specialized facilities or special apartments where they're not going to have very much contact with the outside world. um yeah, it's just so they remain healthy and can get through their bone marrow transplant and their cancer treatment and live a normal life afterwards. So yeah.
Yeah. And can you here, I'm gonna Kirsten, I'm gonna permit you 1 second. There you go. Go ahead and relink if you don't mind. Kelsey, what is so important about having to quarantine when you're going through chemo
Yeah,
and when you're going through a bone marrow transplant? Like, why, if so, next next month in April, I'm going to be able to go home and visit my friend, and I've never met her
yeah,
son. But it's really
yeah,
important that, like, I need to make sure I'm not sick at all before I go. I need to make sure I'm wearing my
absolutely.
mask on the airplane, wearing my mask everywhere in public, because I cannot chance getting that boy sick at all.
Absolutely. Yeah. So with chemo and with the bone marrow transplant, especially in the context of leukemia, your blood system is your immune system.
Mm-hmm.
And so when you're undergoing these treatments to kind of get rid of your cancerous blood cells, a side effect of that is that you also get rid of the healthy blood cells that are remaining. So it's really difficult and it really compromises their immune system because, you're kind of wiping everything out. Um, yeah.
Oh.
And so that's why it's really important to make sure you're not sick and you're not bringing in viruses because the patient's bodies just really don't have a way to fight off any infections that, you know, yeah, a healthy person is in contact with and their body is just like, whatever, we'll take care of it.
Oh, okay. That is, yeah.
Yeah.
No, that's really, really. I'm glad that we're talking about this because it's a
Yeah,
big deal, and I think
it
that
is for sure.
that's something that we, even with COVID, there's people out there who before COVID had these issues, and I think that now after COVID, it's just kind of brought the fact that some people are just immunocompromised their entire life, and it's kind of brought it to our attention.
Yeah. Yeah, for sure.
Um, cool. So tell us about what what impacts does your research have, like if like on finding a cure for ALL.
Yeah, so I work specifically on AML, so it's a different like ALL is acute lymphoid leukemia, AML is acute myeloid leukemia.
What's the
So it's just
difference?
yeah, that's a good question. So they're, at the top of your blood system is the stem cell, and then the stem cell, like the first step of differentiation is myeloid compartment or lymphoid compartment. The lymphoid side of things is a lot of the immune cells. So those are the things that kind of help your body fight off infections and things like that. Myeloid cells become your red blood cells, so oxygen carriers, other things i- down there in that compartment. In terms of like the cancers, they have different oncogenic drivers, so different things go wrong that turn them cancerous.
You said a what,
Um,
on to.
Oncogenic driver, yeah, so like the cancer driver
Oh,
mutation.
okay,
Yeah,
okay.
yeah. Um, sorry.
Oh, no, no.
Yeah,
I use jargon all the time. And then I
yeah,
have to
yeah.
catch myself because I like it's that's our job. We talk in jargon, and part of good psychom is
Hmm.
de-jargoning it. So, yeah.
Yeah, for
No, it's
sure, for sure.
plus it's good to learn new words.
Yeah. Um, so generally, I think in the context of AML, which are the types of cells that I'm researching and studying, it has the potential to identify new things that we can target with a drug or chemo to
Mhm.
improve outcomes for patients.
Okay.
Yeah. Or the potential to identify something that's a biomarker for a more aggressive or less aggressive subset of leukemia, and that can inform or the treatment strategy for the operators as well.
Oh, cool.
Yeah.
Guppy says there's a simple way to think about it.
Yes.
Oncogenes. So our drivers. So oncogenes. Those are genes that switch the cancer on and off, basically, or
Yeah,
alterations
yeah,
that are.
that's a good way.
Oh, okay, cool. We have another donation from Not the Brain. Thank you. Oh my goodness, thank you so much. And we have a raffle. Do you mind if we do a raffle real quick? Because we've raised over a hundred
Yeah,
dollars. I'm really excited.
let's do a raffle.
Cool. So we have X. I'll do a raffle. We're gonna raffle off for a holographic volcano sticker to thank you guys so much for the donations. Exclamation point join to enter, or exclamation point raffle. It'll go off in about 4 minutes, and we'll learn who wins this sweet holographic volcano science sticker. Yeah, sweet. So one thing I wanted, and thank you guys so much. This has been an amazing stream already so far. I wanted to ask you, like, you're talking about these oncogenes, right? What
Mhm.
are things that can, like, you hear smoking causes cancer, right? Or, exposure to this causes cancer. You go to California, they have a Prop 45 that tells you that everything you even think about causes cancer, you know?
Yes.
So uh, I think, like, how does that happen? How can things that are that we're not born with, right? So some stuff, you're born with, and you might get diseases through genes that you have your entire life, and then other
Mhm.
ones, you become at risk of cancers through things that you do or environments you're in. How does that work?
Yeah. Great question. So you're totally right. Some cancers, you're born with the gene that is going to eventually give rise to cancer, which is, sad. It's another line of research that people are working on, trying to create interventions for those types of cancers. In terms of like environmental things you're exposed to, a lot of the times it works. So if you're exposed, to use the example of cigarette smoke, because I think a lot of people are really familiar with that, it is, there are a lot of little chemicals that are coming into your lungs in this case, and they're introducing mutations into your DNA. So instead of making the correct protein to do a certain function and then turn off or be degraded or, go away somehow in your cells, um, these mutations can happen in proteins or in genes, and it can lead to an uncontrolled cell growth. So yeah, so your environment is introducing mutations into your genes, and then your genes are not performing their correct functions, and that is what's leading to the cancer a lot of times.
How is there a certain amount of exposure that somebody has to, or I guess, is there a certain amount of the toxic, I don't know what to call it, like environment or environmental factor that somebody has to be exposed to in order to trigger these changes? Does it vary by race or gender or anything, or socioeconomic class?
Yeah, I'm not sure if I could speak to variations by race or gender. I think yes, in like generally there is a limit. So like, your genes are, we're exposed to things all the time that are causing small mutations in our genes. And usually your cell has machinery that's going to repair those mutations so that you stay healthy and your cells continue to do their proper functions, and you don't have cancer, and it's great news. Um, if you are if you're exposed over and over and over to mutations, so like if you smoke for 20 years, or if you're out in the sun for 8 hours a day and never wear sunscreen, then it becomes really hard for your cells to keep up and keep repairing those mutations that happen. um, and that's when you're at risk for developing things like cancer.
Okay, so it has to be like
Yeah.
so much that your cells just can't keep up, and they're just like, I can't.
Yeah. Yeah. And
Oh.
then, yeah. And so there's a big environmental component of it. Another reason cancer develops is because of aging. So I like to think of it, you start your work day and you're really on top of things, you're making no mistakes, you're catching the mistakes you do make. But as let's say it's 4:45 and you might see you're making mistakes that you're not catching, and you're, messing up things at work. And that's kind of like what happens over your cell's lifetime, where, when you're young, it's really easy to divide. And if you make a mistake when you're dividing, you can fix it. But as you get older, your cells kind of get worn out and then make more mistakes and are less able to catch them. And then you can develop diseases that way. Not even
Okay.
like environmental factors, just like
Just
an innate
age.
biological factor. Yeah.
Wow.
Yeah.
Wow. That's really really interesting. So, one question that I know I really wanted to ask you, and Guppy, I'm really glad you're in chat. Oh, great question about
Yes. I'm glad
ethnicity.
Guppy's here too.
There's social determinants of health that can affect someone's risk. Food deserts or proximity to factories due to redlining. Ooh, that's good. So, how how would something like that, like being in a food desert, food desert somewhere for anybody in chat who doesn't know, where you don't have access to healthy food in your immediate areas. So if you don't have a grocery store but you got $12 generals in a Big Lots, that's a food desert. And those typically do happen in lower income communities. Um, and so I'm assuming in that case, Kelsey, would you be at greater risk for cancer because of inadequate nutrition?
I think it's definitely one of the lifestyle and environmental factors that can contribute to it.
Okay,
Yeah.
cool. Yeah,
Yes. So Guppy says we know that ultra
ultra
processed
processed
foods
foods increase the risk
increase
factor of
the risk
cancer. Wow.
factor.
Wow. It's amazing how much
Yeah.
we've learned in like the past just like 20 years. Because I remember in like the early 2000s and 90s, which I'm aging myself and you're probably way too young. But like everything was like fat free, super, like ultra processed foods were the most amazing thing because you could make them healthy and fat. And now we know that like that was terrible, terrible plan.
Yeah.
Um,
Yeah. And it didn't even taste good either. Like,
there's those little Debbie snack cakes. I mean, I gotta
yeah.
those little potatoes at the bottom of the food pyramid. There we go. Cool.
Yeah.
And it looks like we have a raffle winner, not the brain. You are the winner of our holographic sticker. Congratulations. Congratulations. You can put in your address in the link at the bottom or at the panel at the bottom, and I will send you your sweet hollow sticker. Thank you guys so much for all of the support. Really, genuinely, it means a lot. Um, so what is something like when somebody undergoes chemo, what happens? What do they go through? What is chemo, and then what's physically happening to the cells, and like how does that make the person feel and stuff? Like how does chemo actually kill cancer without killing the patient?
Yeah. So let's see. It's kind of so much in the way that like mutations can cause cancer. If your cell senses that it has too many mutations in it, it can undergo a programmed cell death called apoptosis. So the way that a lot of chemotherapeutic agents work is that they'll enter the cell. Usually, it's targeted by some, you know, factor that the cell has, so we can like direct it to the cell, and then it's introducing just a ton of mutations into the cell to the point where the cancer cell senses like, whoa, there's a lot going on here. We need to do apoptosis. We need to do this programmed cell death. And so that's how it works. So it's kind of like weaponizing a cell's intrinsic
Oh.
factors against it. And there are other things, other types of chemos or drug treatments that target things on the outside of the cell. So, like the receptors we were talking about earlier, sometimes those receptors are mutated in a way that makes them constantly pumping these growth signals into cells. So you can target those mutated receptors um,
Hmm.
and get the cells to stop growing and die that way. Um, and I think getting it to be targeted is really challenging. And I think that's why a lot of the times, patients have really severe side effects from chemo because there are some like off-target interactions with your healthy cells. Um, yeah, so that's why you feel so terrible when you're undergoing treatment.
Oh.
Yeah.
Okay, MedBibs have is a great question. I have been told I have an increased risk because I have a genetic mutation from birth. What types of cancers are those as opposed to cancers that you know develop as a result of these like biological or environmental changes?
Yeah, I think it really depends on the cancer subtype. So I think I can't speak to all cancers. Cancers is a very
Very
um
wide, varied, and
general
changing. Yeah.
term for like diseases that are honestly really different depending on the systems that they show up in.
Oh.
In terms of blood cancers, so ALL is more common in children because it's oftentimes due to something like a genetic risk you're born with, whereas AML, like the disease that I study, is more common in adults because it is a result of like these acquired hits and mutations that happen.
Okay. But so do adults can adults develop ALL, or if somebody has
Yeah.
that gene will they get it as a kid always?
Um, I I'm not sure. I think adults can develop ALL. It's just a little bit less common. Maybe Guppy can speak more to that.
Um, hang on. Yeah, Guppy, do you know? And not the brain. It's below chat. So like if you go, it's scroll down and like the about me and all the panel thingies and the one of the volcanoes. But yeah, that's where you submit the link, or you can send me a whisper. If I think I might already have your address, but I'm not sure. Um, Sorry. Thank you so much for answering that, Kelsey. I really appreciate
Yeah, of
that.
course. Of
So
course.
with AML, do children not get that then?
Children can get that. But it's a little bit less common than with adults.
Okay.
So I think it kind of speaks to the heterogeneity of the blood cancers in general.
I agree. I agree.
Yeah.
Thank you so much for the gift sub, DL Miller. I really really appreciate it. So what's a blood cancer? What is that versus say like breast cancer or lung cancer? What is the difference between a blood cancer and other types of cancers, and how do you fight it?
Sure. I have another picture for you guys. I just oops, sorry, just sent that to the Discord.
No worries.
Um, so with regular cancer types, or like solid cancer types, those are characterized by the development of a tumor
Uh huh.
times. Um, yeah, so you'll find a lump or a lesion and know that that is cancerous. With blood cancers, they're characterized by a buildup of a certain cell type. So I don't know. Do you see the bone
Uh huh.
marrow image that I just sent you? Okay, is it displayed?
Yep.
Okay, great. So on the left side with the healthy bone marrow, you can see that there's a lot of different types of blood cells in it. Whereas on the right side in this AML leukemia state, you can see that there are a lot of big purple, like purple cells.
Mm-hmm.
Um, and that is the tumor in that case. Like that is the cancer
Oh.
cell. So yeah.
So to get rid of this, do you have to like do they have do people have to deal with blood transfusions and stuff? Do they need new blood? How how do you get rid of this?
Yeah, so you can get rid of it in a couple different ways. A lot of the time, people are treated with chemotherapy agents, so that will target these cancerous cells and hopefully reduce the burden of the cancerous. They're called blasts in this case. Another thing that can happen is a bone marrow transplant. So that is basically getting rid of all of the cells in a patient's bone marrow, or most of them that are unhealthy, and then introducing new cells to repopulate the bone marrow that are healthy.
Oh, how does that
Yeah.
so about how long does that take, and do adults and kids have the same like treatment timelines?
Um, Yeah, so it depends. So I know you asked me about that on Tuesday. So children tend to have a longer treatment timeline, and that's because adult leukemia clinical trials and pediatric leukemia clinical trials have just happened in completely separate worlds for a long time.
Hmm.
So yeah, that's so
Does,
that's one reason.
is it just like
Um,
adult cancers are more researched? Is that it?
I don't think that's it. I just think like the doctors are different and the clinical trials are different. So the treatment strategies that they found are different.
Oh,
In general, like children have longer treatment um, timelines because children are a lot more resilient and can handle like higher or longer doses of chemotherapy than adults a lot better.
really? I would not
Yeah,
have expected that. I would have thought the opposite.
yeah, yeah. It's kind of crazy, but when your body is that young, like your cells have a lot more, I guess like regenerative
Uh huh.
capacity is how I would put it.
Does that mean that, sorry, excuse me, since they're more resilient and can handle more chemo, but does that also kind of mean that the cancer cells can come back easier?
Um, I am not sure about relapse rates in children versus adults. Um, yeah, I'm not sure. I think part of the reason that the treatment courses are longer is to prevent relapse rates, but I'm not sure how that differs with children and adults.
Okay. Okay.
Yeah,
And then I'm not sure if you know it or if you know the answer to this or if Guppy does in chat.
yeah.
Are are doctors concerned about like a kid's future development as a result of chemo, or like is it you know how do we know if chemo affects their long-term growth? Is that something that doctors are concerned about, or is it genuinely just that like kids can deal with this better?
Yeah, I think that's definitely like always a concern is how the long term or what the long term effects of your treatments are going to be. I don't think we know a lot right now, partially because, leukemia patient outcomes have really improved in the past couple years.
Oh,
So
good.
prior, to 20 years ago, there wasn't a huge cohort of patients that you could follow up on for their entire lives, just because it was such an aggressive disease and the treatments just weren't there yet. But now I know at the Hutch, the case that Baker Lab is doing long-term follow-up treatments for pediatric leukemia survivors. So we're really working to kind of characterize some of those long-term effects and look
Cool.
for, this person underwent a long leukemia treatment as a child, so maybe they're more at risk of X, Y, and Z conditions.
How do you see like that research in 20 years? Like what do you think, or what do you hope will be possible in 20 years? Because I know you want to say like, oh, we'll find a cure, but we know we're scientists and we know how long research takes and where we're at right
Right,
now. So
right.
what do you think? Hopefully, we'll be a Nobel Prize winning biology laureate and study cancer cells and have amazing research findings. But what do you hope will happen in 20 years with the status of leukemia research?
Yeah, I mean, I really hope that and I guess I can speak to my field because I know kind of better like the state of my
Oh
field.
yeah, no, just
Um,
to what you can speak to.
yeah, yeah. So with AML, the relapse rates are really high for adult populations that are, undergoing treatment. So I hope in 20 years we have identified things to target to kind of prevent those relapse rates, you know what I mean, and improve outcomes in that space.
Cool. Thank you so much
Yeah.
for the gift sub, Anonymous. I'm not sure who you are, but I know Kelsey appreciates the gift
Oh,
sub, and I do
yeah,
too.
thank you.
Enjoy your sweet emotes. So when when somebody has cancer and they're, let's say, I know Well, you're most experienced with adults with AML, so let's go with that.
Yeah, sure.
What happens to the cell? Like in the cell, what changes? I always like to give people a, you can talk about like, oh, I researched cell changes in cancer, but this is the experience where, we want to know the nitty gritty behind the scenes.
Yeah.
How do things work?
Yeah. So I mean, this is I guess we were talking about this more generally earlier, but in AML there are like a bunch of different driver mutations. That's why it's so kind of challenging to treat sometimes and so relapse prone. But basically genes can become mutated or they can become fused to other genes in a way that uh, is bad for the cell. And all of these things, no matter what they are have the end product of making your cells uncontrollably grow and proliferate and do that faster than your healthy cells so that they overtake your your bone marrow and your blood system. So,
Okay, so there's no like with the blasts that you showed in the let me find a there again. So here in this photo that you've got with the AML and you've got the blasts,
mhm,
are like, I guess what I'm asking is more of, sorry, I didn't phrase my question quite right. I guess what I'm
sure.
asking is more of like, so in 2019 I was diagnosed with like pre-cervical cancer, and I had like stage
Mhm.
3 CIN. And the different stages of that is, it goes like a third, 2/3, then all the way through the cell, like the changes. You knew about this, Sushi. Um, so I guess kind of that's what I was thinking about, where like, different percentages of these cells have mutations. Is that how it works with AML?
Um, Kind of. I think it's more like AML is staged by um, determining what percent of like the bone marrow or the blood is these leukemic blasts.
Oh,
Um,
okay.
yeah, yeah. But I'm not sure if there's, I don't know, like, a certain percent of cells have undergone these changes. I think it's just quantifying the number of cancer cells.
Oh, okay. Oh my goodness.
Yeah.
Please tell me this is not what's going on. No. Okay, cool. I'm like literally in a Slack, and we've been waiting for 4 days for an eruption to start in Iceland, and everybody is
Oh
on
no.
standby. And I'm like, oh my god, my Slack is beeping on a Saturday. That's not good. Um, cool. You don't think there will ever be a cure, med dibs, but diagnoses, better management, oh, okay. So like, you think that there will be, yeah. What are ways that people, let's say if you have been through chemo, right?
Mhm.
What it's exclamation point Fred, Sushi. It's exclamation point Fred. So when you've been diagnosed with chemo, you've gone, or with cancer, you've gone through the treatment, and like, what is considered successful? What is the end goal with that one round? Um, and then, yeah, I guess how can people prevent relapse, or is there a way?
Yeah. Yeah. I'm not sure if they've found a way to prevent relapse in AML. I think the goal of the first round of treatment is always to achieve remission status. So that's like, if AML is more than 20%, I think it's more than 20% of your bone marrow are these cancerous blasts. Remission would be less than 5% are cancerous blasts.
Okay.
And then you do maintenance chemo, or there might be a stage between those two. But you continue to do treatments to hopefully wipe out those remaining populations of cancer cells.
Until you're down to below 5%, or is the maintenance chemo
And
after
until
5?
they're after 5%, so until to get that number even lower.
Okay.
Yeah.
What if it comes back? Is it classified as a relapse once you hit above the 5% threshold again?
I'm not sure how they classify relapse. If
Okay.
Guppy is here, maybe he knows more of those details.
Okay. I'm sorry. This is why my teachers didn't like me. This is
Yeah.
why I'm a scientist.
No, I think these are like great questions. I just don't know.
No, it's fine. I like
Yeah.
doing coffee with a scientist for this reason because I'm
Yeah.
always interested in learning more about different, you know, disciplines, but I don't know enough, you know?
Yeah, for sure.
Um, I think I wanted to talk a little bit. Could you speak to when and I know you don't deal with the families as much. You were definitely on
Right.
the research side.
Yeah.
How does your research translate into what a family coming to Fred Hutch for help with a cancer diagnosis receives? Like, how does what your research does right now translate into the care that they receive? Or, like, how does it correlate?
Yeah. So the research I'm doing right now is definitely like, you know, very early stages. So it's probably not going to manifest in the clinic for decades, right? Um, but right now, research like mine has really informed treatment strategies, and it's really informed the way that we classify different leukemias and the way, you know, and how we, it's given us better predictions of outcomes, better predictions of how well the patient's going to respond to chemotherapy, and things like that. And it's led to some drug development, but that all of those things take a lot of times, and I am just in the very, very early stages, so it's going to take a while before that happens. I think you might be muted again.
Oh, I said, I know that's a long time, but you
Yeah.
know you're making a difference.
Yeah, yeah, definitely.
Oh,
um
we have
yeah.
another donation. Let's see who it is. Not the brain. Oh my goodness. Thank you. Thank you so very much. Really appreciate it. Definitely helpful. Absolutely helpful. We're getting to almost $150. I'm really excited. Um, could you tell us a little bit more about what families who might be, they're coming to Fred Hutch to deal with this? What do they have to do, or are you familiar with that process, like when
I
somebody's coming to get care or help?
Yeah, I'm really unfamiliar with that process, honestly,
Okay.
just because I sit on the research side of things so often, and I've, very fortunately not had to support a family member through a leukemia diagnosis yet. So I don't know. I'm sure like Kirsten or Guppy would have links and resources to
Mm-hmm.
some of
Cool.
those things.
Have you ever thought about what you want to do after your PhD?
Yeah, so after my PhD, I want to go into science writing or communications or some aspect of that. So really excited to be on the stream today
Yeah.
and getting to kind of explore how I'm going to communicate this research and getting some practice doing it as well. Um, yeah, I think I got very interested in that, classically because of the pandemic. And, I don't think I think there's a lot of mistrust in science right now just because a lot of people don't have access to scientists and an ability to ask scientists like all of their burning questions. And yeah, so I really want to kind of help there and rebuild some of that public trust.
Speaking of which, chat, if you have questions, this is the time to ask. We have a
Yeah.
real scientist here. Here's a link for Guppy. I'll permit you, and it should allow. Or you can send a link to Sushi, and he will post it because I have links disabled for safety reasons. Um, yeah, no, I appreciate it. Sushi can do it, and then yeah, he'll post the link. So, what is something that you can tell us about your so far grad school experience? This is something I always love to ask people. Excuse me. What is the craziest, however you define that, like
Yeah.
research experience you've had? Have you had like a lab mutation monster crawl out of your fridge, or like, I don't know what happens in a bio lab. Like what's the
Yeah, I know. I know when we met on Tuesday, you asked me this, and I'm like, biolabs are so relaxed, you know? There's nothing, maybe not
really?
relaxed isn't the right answer, but I always joke, like my job is pipetting clear liquid into another clear liquid and seeing what happens. So no mutation monsters yet. Fingers crossed we will never have them. But I think like the craziest day I've had in lab is we had this very large, very intense study that we had to do. So my coworker and I worked until 12:30 in the morning, like trying to get this experiment done. That's only happened once. Like this is a regular thing we try to do, but it was very weird to see like all the happenings at Fred Hutch at 11:30, 12:30 in the
What's
morning,
going on? Like I've pulled my, overnight shifts in a lab,
Right,
and it's like you and the
right.
security guard.
Yeah, it's the security guard, it's the like night janitor who was just as scared to see me as I was to see him. I don't know. Red Hutch has a very like it's really nice. Like all the core facilities are like underground, and so all the buildings are connected by tunnels in the basement.
Oh, you don't even have
Uh,
to deal with the rain.
yeah, it's really nice. It's really fantastic. But it's also a little spooky to be walking through the basement tunnels at night, just trying to get to my coworker's car to go home.
Yeah, I
Uh,
that sounds really creepy. That
yeah, it
sounds
was
really creepy.
only happened once though, and
That's okay. I mean, there's still plenty of time.
in the tunnels, like
That would have been nice. We had a mountain lion escape on campus one time, and I was like trying to
what?
walk home at late at night, and yeah. So we put out like, I like called my husband. I was walking. I was like, I need you to be on the phone with me this entire walk home because there's a mountain
In
lion
case
on
I get
campus
mauled, yeah.
somewhere. I need somebody to help me.
That's crazy. Where is your campus?
I was at University of California, Santa Barbara. But the
Okay,
mountains are, so we have like the campus is right on the beach. Which, dude, try to go. It's really clutch. It's beautiful. But
okay.
yeah, no. So the mountains are very very close, and there's like mountains, a little strip of land, and then ocean. And boy, if they're looking for food, they just come right down. And there's plenty of,
That's crazy.
fish and seabirds and stuff. And they had just trotted through campus to try to get to the estuary for lunch or supper.
That's crazy. That I wouldn't have left. I would have just camped out, you know?
No, I really wanted to go home.
Yeah.
Yeah, really wanted to
Oh,
go home.
well,
We have some great questions in chat, and thank you so much,
yeah.
Guppy, for sending this link on how AML treatment works. I appreciate it.
Yes.
Sushi wanted to know, I don't know a better way to ask, but what are some early signs you should see a doctor if it could turn out to be cancer? I realize this could be super broad.
Yeah, I don't so I know for leukemia the big one is just like for myeloid leukemia your bones will hurt and very easy bruising will be happening and you'll just feel like generally bad and tired. I can't really speak to other types of cancer. I'm not a trained clinician.
Yeah,
Like these
you
are
study
just
what you
things
study.
that, yeah, I study what I study. These are, the myeloid leukemia symptoms. Those are just things that I've read. Um, but yeah, I'm not sure.
Okay. No, not
If you're concerned, go to a doctor. That's my official stance. Yeah.
always good. Always good.
Yeah.
That's nothing. I've spent 72 hours playing games. Okay, it's important work. I'm not doubting that. I am not doubting that. DL Miller, my dad's biological mother passed of pancreatic cancer at the age of 54. As I understand, also had cirrhosis of the liver from prolonged use of alcohol. I'm wondering how much of a factor does alcohol play in the development of cancer and the body's ability to fight it, etc.? That's a fantastic question, DL. Thank you for asking.
Yeah, that's a really great question. I don't know if I could speak to alcohol's effects to cancer specifically. But I am thinking back to this biology or biochemistry professor I had in college who was like, alcohol is a toxin, and the reason you all come in with hangovers is because you're poisoning your body, and you're having to your cells are having to work really hard to clear it and to um, return your body to baseline.
Oh,
So
that's what's
prolonged,
happening.
yeah, yeah. So that's why it's so bad for your liver, because your liver cells are the ones that are actually like doing the processing and yeah, and having to like work overtime to get rid of your hangover. Um,
Oh.
so yeah, that's but I can't speak. Guppy just said, unfortunately, increased alcohol consumption raises your risk a lot,
Of liver and pancreatic.
particularly of liver. Yeah. So
Woof. Woof.
yeah.
What are some other things that you know that can think of it as your body is working out while you're standing still or curled over or staggering? That's a really good analogy, Ningao. What
Yeah. Yeah.
I know I'm a little bit curious. You've been telling us about these clear fluids. Could you, if I know, like sometimes with science and when you're working in labs, you do have to be confidential and things like that. And I know sometimes I'm even cagey when talking about ongoing research because you never know who's out there
Mhm.
watching. Um, which is why I'm explaining this to Chad. If we've seen a little cagey, that's why. That's why because you could
Yeah.
get scooped. And also
Yeah.
sometimes like your PI who runs your lab might not want you to talk about certain things. So if we're
Yeah.
seeming a little bit cagey, that's probably why, and we're very open about that. Um, what what are you doing in your research? Like when you're piping clear fluids and looking at cell changes, can you tell us about your experiments?
Yeah. So my experiments have really evolved since I've joined the lab, as there's a saying like you've got to follow the science. And so I've been following the
Mm-hmm.
science and it's kind of changed what I'm doing. A lot of what I'm focused on now is that we've found that some of the mutations that I'm introducing into my cells, so some of the things that I'm interested in studying, are contributing to some chemo outcomes for cells in a dish. So yeah, so a lot of
What
what I've been doing is,
what is that? If
sorry,
if you can explain more, if you can,
yeah,
what does that like? You're causing cellular changes. You're causing changes in
hmm
cells, and then are you causing different types of changes in treating with the same chemo, or the same changes in different chemo? And what?
yeah. So I'm causing like a known change in what I'm interested in studying, and then I'm treating cells without the change and cells with the change with chemo, and I'm seeing that the cells have different outcomes when I change them. So
So the cells that you've treated with chemo,
yes,
the different chemos have different outcomes, or the same chemo can have different outcomes?
it's the same chemo can have different outcomes depending on the attributes of the cells. So
Oh,
it's been really exciting thing to find this early
is now I'm really is it different types of
on.
like is it like a pancreas cell versus I don't even know how like cells. I'm so bio not inclined.
Yeah, yeah.
Is it like a foot cell and a lung cell, or is it like two types
No,
of the
they're
same cell, or
yeah, two like they're both AML cells and I've introduced a change into one of them and that like the singular change is causing a different outcome, which is exciting.
oh, cool.
Yeah,
That's
yeah.
really really cool. Do you think like Yeah, I don't even know how I imagine like what's the next step? You found this out. Like e- eventually
Yeah,
something like
so
this would go to human trials. What's like the how does that process work? How do you get from something that you as a researcher do in the lab to like a human trial to see if these drugs
yeah,
really work for people?
yeah. So it's super challenging. I think what I'm working on now is I'm trying to figure out why. So I know I've caused this change. I know I see this different outcome, but that's A to, J, and I'm trying to find all the different letters, as to like why we are seeing these different changes, like what's going on in the cell, like are there different signaling pathways involved? Are there different um, like yeah, why is the response so different? So that's step one, and that's probably going to take years. um,
Oh, years.
yeah, yeah, and then
Why does it take so long? Because I know a lot of people don't get how long it takes. So why does it take so many years for this?
just because like science is hard and biology is really complicated, and your cells are really amazing at changing and reacting to things. And so trying and sometimes it's not one change, sometimes it's like five changes, and you have to figure out all five of them,
Oh
kind of,
man.
you know what I mean? And there are like tons of different signaling pathways in your cells, and they're all interrelated, and they can all, you know, there's like canonical things that are well established. There are non-canonical things that have come about in the past few years. And you have to consider all of them when you're designing these experiments so that you can really be able to say, it's this thing or it's this list of things. And I know that because I've done so much work to figure it out, you know?
Okay. So like, and thank you so much for the follow, Buckvac. Welcome on in. And anonymous, thank you for gifting that sub to Kirsten at Fred Hutch. We are raising funds for cancer research, and we have Kelsey Woodruff with Fred Hutch here. She does scientific research on AML, which is a type of blood cancer, and we are raising funds for that sorts of research. So if you're interested in donating, please do. We would greatly, greatly appreciate it. Even just $5 can help buy 100 test tubes, which, science is pretty expensive. Really, really, really
Yeah.
expensive. Have you, like, how did you come to work with Fred Hutch? Did your PI have a connection? How does somebody go from being a, biology grad student to working with a huge cancer research center? Like that's a
Yeah.
great position you're in, How did you
Yeah,
come to
yeah,
do
it's
this?
it's super exciting. I love working at Fred Hutch. That's, obviously I wouldn't be here if I didn't love it. But one of the things that really drew me to my program is that it's collaborative between the University of Washington and Fred Hutch. So when you join the program, you have the option to rotate and potentially join labs that are affiliated with the university or labs that are affiliated with Fred Hutch. So I chose to do some rotations at the Hutch and eventually decided to join and do my dissertation here. So
So does that
yeah.
mean you're PhD advisors with University of Washington, but your PIs are at Fred Hutch?
So my PI is at Fred Hutch, and she's also my advisor. So she has a dual like
Appointment.
affiliation.
Okay,
So she's
okay.
yeah, yeah. So she's
Cool.
affiliated with like the pathology department at UW, I think, and then she's also affiliated with a department at Fred Hutch, and the lab is physical.
Okay. And pathology, what is that the study of?
Oh gosh, I don't know the exact definition. But pathologists can really look at like I guess the images we were showing earlier. I'm not sure if they're
Oh,
still up.
like
I
the
can't see
blood,
the
they
full
can look
screen.
at the blood cells and tell.
Yes. Yeah, and
Oh.
there. extremely gifted. I've been in lectures from pathologists and they show me two things that look exactly the same to my untrained eyes. And they're like, and you can see this patient has this form of disease that, we can treat with X, Y, and Z. And I'm like,
Corporate
that's
says
an
that's the
amazing skill
same
set.
picture. Like,
Yeah. Yeah. So I don't know. It's a really amazing skill set in my opinion.
no, that is. We said, Guppy says a lot of Fred Hutch faculty have cross appointments at UW, and it's the adult oncology arm of UW medicine. Very cool. That's really cool. So when when a student if a student wanted to go this way, if somebody wanted to pursue this path,
Mhm.
what would you recommend they take in undergrad?
Oh, well, I guess, my recommendations are going to be based on what I did,
Well, yeah.
right?
Or and also like because I know I feel kind of this way about some things. Is there anything you feel you didn't learn in undergrad that would have been really helpful to you
Mm,
now? I like to ask these
yeah.
questions because I know, like, man, if I had only known about project management, oh, so I like to tell people, like, yeah, do this, don't make my mistakes.
Yeah, yeah, for sure. So I did my undergrad in biochemistry, which I feel like classes wise really prepared me for graduate study. The thing that I think is almost more important if you want to pursue research in like a PhD program or undergraduate school is to really get involved in undergraduate research and get some like hands-on doing real science experiment in a lab. Because you have lab classes, but a lot of those are like cookbooks. We're gonna isolate the DNA and then we're gonna look at it or do something that we know is going to work just to teach you how to like do the technique.
And then here, write up a
um
lab report, and there you go.
yeah,
Yeah.
yeah, exactly. But when you do undergraduate research, your experiments aren't going to work all the time, and I think first of all learning to fail and just like get back up and not view that as a reflection of yourself is really important in undergraduate. And then it's also important to develop the, okay, well, I saw this thing that I didn't expect. What could be causing it? And really thinking through all of the different things that, and how you can test to see which one is actually going on.
Okay. Cool.
Yeah.
Um, have you ever met anybody when and you're working with Fred Hutch? And I'm gonna plug
Hmm.
here a little bit because I know that excuse me, Kirsten's been dropping facts in chat, and one of the reasons Fred Hutch is so big is because it has a decades long history of researching cancer, like uh, let's see, COVID, HPV, which is the virus that causes cervical cancers, like the one that I had, AIDS. Does AIDS link to cancer?
I am not sure,
Okay.
frankly. Yeah, I think that's kind of like a separate arm.
Oh, Guppy says yes. Guppy
Yes.
says
Okay.
yes. Pretty much everything, anything that can do harm to cells. I think that's kind of what I'm learning today. I'm learning
Yeah.
today that like pretty much anything that can change a cell, even just getting older, can help contribute to cancer.
Yeah.
This is a very strange question coming from somebody
Sure.
who's lived for a decade in California. Does everything really need the Prop 45? This will give you cancer warning, or this can give you cancer warning. Like, is that like
That's a good, good question. Um, in my opinion, probably not. Just because, something that I'm constantly amazed by is just like how resilient your cells are. Like you're throwing a million different things at your body every single day, whether that's like the things you're eating, like sunlight, what you're like breathing in when you go outside. And your body is really amazing at adapting and dealing with those stresses at baseline. And so, it's like everything can cause cancer, but some things can cause it like cigarette smoke, for example, like that, okay, there's decades of research to establish that cigarette smoking can cause cancer. And a lot of in my opinion, like the things that are randomly labeled, like, oh, this increases your risk of cancer. It's like, yeah, like if these dozens of things like happened and it was like just the right time, like, yeah, like, it could introduce a mutation that causes cancer, but it's probably not going to. Which I think is like a nuance that's missing from a lot of those labels.
No, I'm really glad that you
Yeah,
said that. I'm glad. I'm
yeah,
glad because I feel like when somebody asks me like, "Oh, will this make the volcano erupt?" I'm like, "Well, you kind of have it's the same. It's the same. You have to have all the factors already ready. You're like almost there, and then
hmm
something can tip it over the edge." And like, like you said earlier, years and years of cigarette smoke or years and years of some exposure to some toxin can cause those changes, but
right,
for just one thing, it's already kind of got to be almost there to get tipped over
right.
the edge.
Yeah,
Um,
exactly.
wow. Wow. So, have you ever gotten to meet anybody like higher up at Fred Hutch or anybody who's like, you know, I don't know, made an amazing discovery?
I mean, I think everybody at Fred Hutch is doing amazing work and like
That's
making amazing discoveries. Like I think it's a very prolific research environment. Like and just like the level of like creativity and um like resources that the center has to like really push this novel research forward is it constantly amazes me.
mm-hmm.
Um, but so yes, in that like I think everybody is doing really amazing work there.
Yeah.
Um, I had the opportunity to work with Harmeet Malik, who's like a big name in the genetics field.
Ooh.
And although I decided I didn't really like that field very much just because it wasn't the science I wanted to do, like working with him was really amazing. And I got to kind of experience science. Like his lab is really big, so experiencing science on a scale that like I've never experienced it before was like really amazing too.
Yeah, I couldn't. That's insane. I couldn't imagine being in like a big lab. That's
Yeah,
wow. Wow.
yeah.
DL says, on the one hand, I agree, but on the other hand, with the litigious way our society is set up, if it's not labeled, it opens the floodgates for lawsuits. Yes, I understand that. I get it from that
Yeah,
point of view, DL. I will
right.
agree for that point. I'm just thinking like, will I really get cancer if I stay at this Days Inn? I mean, okay, well, will I really get
Like it's
cancer if I
not
stay at a Hilton? Because like Days Inn, no, I'm kidding. But
right.
yeah, it's not like that.
It's not worth having the constant anxiety about it. Is what I was trying to
Yeah,
convey.
I agree. I agree.
Yeah.
If you want to read more about one of the amazing scientists, Dr. Fred Applebaum released a book last year about the pioneering bone marrow transplants done at Fred Hutch. That's awesome. So Fred Hutch
Yes. That's a
pioneered the
really
bone
cool
marrow.
book. Yeah. Yeah.
Wow.
The first bone marrow transplant ever happened.
How did, or if you know, and I don't know, how did they come up with this? How did they realize that, bone marrow transplants were the way to help to get rid of leukemia?
I'm not sure, actually. I don't know. I mean, it seems like one of those things that's kind of straightforward now, like looking back, it's like, okay, like, it kind of makes sense that if you have this disease state in your bone marrow, you could, in theory, get rid of it and then reintroduce, new cells that are healthy and that would cure your cancer. As far as like that makes sense to us now, but like back in the day, I'm sure that was like a completely zany, like, nobody had ever heard of it before idea. So
Wow. Was it
I'm
like
not sure. I think
I'm like genuinely asking. I'm assuming like this
right,
made like
right.
national news and everything and was just like a
Oh, I'm sure. Yeah.
breakthrough. Wow.
Yeah.
Do you know how so Fred Hutch opened in 1975? And do you know about how many people have had bone marrow transplants at Fred Hutch since then? Because it was the first bone marrow transplant in 1975.
I am not sure when the first book.
Okay.
And I'm not sure the number at Fred Hutch, but over 1 million people have received bone marrow transplants just across
Oh,
the
wow.
world.
Wow.
Yeah, so it's been a really impactful development in the treatment of cancer.
Oh, wow. So 1950. Wow, 1956. I like,
Yeah.
do you feel like you could have been, okay, like gender
Yeah.
aside, because like we wouldn't have been allowed to be scientists, but gender
Right.
aside, like I couldn't even imagine being a scientist without the internet in 1956. Like,
Oh my
how?
gosh, I know. I was talking to one of my coworkers about this like a couple weeks ago actually. And just because like, scientists are very prone to overworking, and I think one of the reasons for that is because like, science moves slow now. It moved like glacially before, you know, like in the past. So, it just even things like reading literature took a lot more time because you had to physically go to like a library and like dig up all these sources and synthesize them and process them in your brain. Whereas like now I have like 10 tabs open right now of papers that I want to read or that I have been reading that are informing strategies for what I'm going to do next.
Did
So,
did you and this is a I I'm sure we're all Google Chrome has tab groups now, so you can have
yeah.
20 tabs about the same thing and then condense them into one. So now you can have a billion tabs open.
Yes. Yeah. I am a tab monster for sure.
Yeah,
Yeah.
absolutely. Cool. Well, I know we've got you for maybe about 10 more minutes or so. Um, if that's all right. When it was first
Yeah,
developed,
yeah.
the method was questioned and scoffed at. It wasn't until Don and his team worked out all the parts of the process to allow for repeatable, safe transplants in the 70s that made the treatment mainstream. Oh, yeah, not the brain. Just like click on one and then shift control to click over to the where and then right click and it'll make a new group and you can call it whatever you want. It's amazing. It is definitely going to drain my computer's memory all the time. Yeah. Yeah. Um, wow. So in a bone marrow transplant, and I understand you don't work with clinicals. Could you
Right.
explain how it works? Like, do they literally take out all of your blood, or do you know how that works?
Yeah, so they're not like draining your body of blood or anything. But the way it usually works, from my understanding, of course, your Center Guppy could jump in and fill in some gaps that I miss. But your blood system has to be depleted, so that's usually involves like radiation to kind of like get rid of all of the cells in your bone marrow. Not so much with your like peripheral blood, like the blood in your veins, but the cells in your bone marrow are undergo radiation, and that kills most of them off. So it's getting rid of the disease, and but you can't stop there, obviously, because you need blood cells in your body to do things, have an immune system, stuff like that. So they'll take healthy cells they can be derived from the patient themselves. So you can actually isolate healthy cells from a leukemia patient, or healthy cells from another donor, and kind of grow those up and then transplant them into the body. And they will find the bone marrow and repopulate and hopefully give rise to a healthy system, blood system.
Do How do you target just the bone marrow? Is it do the do you make the chemo drugs so that they only target certain cells?
I'm not sure. So it's less about chemo with bone marrow transplants, from my understanding, and more about radiation that's targeted at
Mm-hmm.
your bone marrow to kind of deplete those cells.
Okay. I guess
Radiation works in a similar way where it's like introducing mutations and killing your cells off that way.
I'm confused because
Yeah,
it's it's hard for me to imagine, like when I think of radiation I think of like x-rays. Is that
yeah.
is that what happens? Is that how they treat this?
I'm not sure like the difference in uh radiation versus like
Okay.
X-ray, things like that. I think it's more intense than just like getting an X-ray,
I,
right? Because you can go get an X-ray and then you
walk
don't
away.
have like a mass, yeah, walk away, like you don't have a mass in cells.
I'm just wondering, like, how does it target just the bone marrow and not the rest of your blood? Like,
That's
that's where I'm
yeah,
confused.
that's a great question and not something I know the answer to,
No, that's fine.
just because I don't work clinically. But
No, no, you're fine. MedDibs wants to know, is cancer a disease or a mutation? That's a great question.
yeah, I think the cancer is a disease that's caused by an uncontrolled buildup of mutated cells. So the idea is that cancer cells have acquired these mutations or changes that are um, overwhelming, that cause this. Sorry, I'm getting distracted by messages in this part. So cancer cells have mutations that allow them to grow faster and kind of be like stronger cells than your healthy cells, so they overtake your body.
Cool. We've got Dana Farber Cancer Institute. Guppy sent me a link about different types of cell transplants, so I'll put that into chat if anybody is interested. It looks like I'm gonna go here. Oh, and there's a Fred Hutch version. Even better. Even better. We're a Fred Hutch stream. We want the Fred Hutch version. Heck yeah. So it infuses blood precursor cells from a donor. What's a blood precursor cell? Is that a bone marrow cell?
Sure, yeah. So they're usually living in the bone marrow, and we kind of talked about it earlier with like the big pyramid of blood cells, where like at the top is this blood precursor cell or stem cell, and that stem cell has the capacity to create all of the different types of blood cells in your body. So all your different immune cells, all your different red blood cells, and it can also make more copies of itself that can, you know, in a similar way, differentiate. It's what the process is called to create your different blood cells.
Wow,
Yeah.
that's really cool. Oh my god. Well, Mandrake, thank you so much for gifting the sub to Not the Brain, and thank you, Anonymous, for the amazing $50 donation. Wait, what? For two? Holy smokes. Oh my god. Okay, we are raffling. We are almost done and we are raffling. We are
Yay.
officially over $200. That's amazing, guys. Thank you so much. This has been amazing to help
Yes,
raise
thank
funds.
you.
This is groceries and test tubes and microscope slides. It does mean a lot. Like, genuinely. We really, really, really do appreciate it. I'm going to put a S raffle exclamation mark join in the chat if you would like to be entered. And since we're almost done, we're going to do two raffles. We're going to do two. We're going to do one for the magnet for raising $200, and then we're going to say farewell to Kelsey and a thank you, and then we're going to do a third raffle just for you guys coming here and being amazing and helping raise money for a cause that is important to, I know, a lot of other people, but to me personally, because I'm like, I wish I could like show this. Dude, this kid's so cute, like stupid
Oh,
cute, like so cute, and he's so sweet, and he's just like the most adorable little thing ever, and he's such a good boy, and he's been through just like so much the past like 2.5 years, and he's
yeah.
just
It's a challenging thing to go through, like it's gotta for him, and I'm sure it's like had a big impact on his support system.
yeah. I mean his family, and I know that it's like his whole family works together to try to make things easier so he can get to his appointments and everything, and
Right.
it's you know he probably wouldn't even be alive if it wasn't for researchers like you and researchers at Fred Hutch.
Yeah.
Even just developing that chemo to help and know, like you said before, that kids are more resilient so they can deal with more treatment. And does that,
Yeah.
does the increased amount of treatment that a child with leukemia gets, is that like, how would that compare if an adult were to do that? Like, what would that do to an adult's body?
Yeah, I think generally adults have a harder time um, recovering from chemo. So I think it would be a more, or I guess speaking for like older adults, they have their bodies have a harder time recovering from chemo. So it would just be a more severe side effects and more a longer road to recovery than for a child. Something that I learned as I was like talking to clinicians trying to find an answer to this question is that uh, adolescent chemo or adolescent leukemia treatments and young adult leukemia treatments are kind of changing to
Oh,
look more like pediatric leukemia results or pediatric leukemia treatment courses. So and people are seeing better outcomes from that. So
and
it's
by
a
adolescent,
changing
you mean like young adult?
like yeah, both. So like late teenager to like mid 20s
Cool.
people. Yeah.
Oh,
Yeah.
that's
So their treatments are changing to look more like pediatric treatments. So kind of indicating that maybe their bodies are more able to cope with chemo than like an older adult.
that's that's awesome. Like that's good to know because
Yeah.
I mean that gives you knowing that at those ages your bodies can be more resilient. You know, if somebody is
Yeah.
diagnosed at those young ages, still it gives them better hope and they don't have to, have as bad repercussions as somebody as an adult. They because they receive a lot more chemo at one time, and and
Yeah.
how does that compare to the amount of treatment if you were to like squish all the time together of a treatment course? I guess what I'm asking is, would overall an adult receive the same amount of treatment as a child, just under a shorter time frame?
That's a really good question. I honestly don't know the answer to that. And I think it would probably vary a lot depending on the patient, the individual themselves, and just like the different attributes of the their types of leukemia. So whether they have like a more or aggressive form.
Okay, so it's all about the cancer. I mean, the
So
the age and everything else is kind of all secondary as to what the cancer is doing.
yeah.
Wow. Cool. Well, before we let you go, I would like to ask you two questions. I would like to
Okay.
ask you if you have any advice on just general, like, what are things we can do to just try to lower our chances? Like, what are just good life advice things that we don't necessarily maybe have to follow because we're not perfect, but like, what would you, in your professional expertise, recommend that we could do to minimize changes in our cells that could lead towards cancers?
Yeah. So not a clinician, big disclaimer, not a doctor. So you totally don't have to listen to me. But I think the big ones that like I follow in my personal life, so don't smoke cigarettes. They're really bad for you. And then wear sunscreen. It's UV radiation causes, skin cancer is very common. And so, um, yeah. And so is lung cancer because of those different environmental concerns. So yeah,
Nice.
those are my big ones. In terms of leukemia, yeah, I don't have any specific recommendations. I think we're all just
That's
doing our
kind of,
best.
yeah, no, I just mean
Yeah.
like Sushi wants to know which sunscreen. I I don't
Any of them.
any of them. Great. Well, I know I'm Buckvac had a question in chat or something. He was talking about the different, or no, it was B Joshers about different like cancers that are sexually transmitted. But I know that that's not with humans. That's with like dogs and Tasmanian devils, right? And that's pretty much it.
Um, yeah, so dogs have transmissible cancers. I think that happens through bites though. I think
Mm.
the human transmissible cancers would be like it would happen because of like sexually transmitted diseases like HPV,
Oh, I see. I see.
things
Okay.
like that. Yeah.
Okay. And then
Yeah.
if somebody wanted to chat to you or talk to you and ask you questions,
Yeah.
are you available on any socials?
Yes. So I will put my Twitter handle in the
You live
chat
in the
here.
PNW.
Um,
You never see the sun. We get sun
I
occasionally.
see the sun for 2 months a year. Come on, guys.
Yeah, it's not fun. We also, congrats, MedDibs, you are the winner. Congratulations, you are the winner of our magnet. If you scroll down, there's some panels. You should click on the one that says send me your address, and I will send you a very cool volcano magnet. Congratulations. Sweet. So you are at Kelsey Woodruff. Oh my gosh, I love that. Is
Yeah,
it
little joke for you guys.
no dash or no slash though? Just at
No, yeah,
Kelsey
I just
Woodruff.
did that to, yeah,
I love that actually. I really
yeah,
like that. That's so cute.
yeah. So yeah, if you guys like, I think my DMs are open. I can make sure they are, or just tweet at me. I'm happy
Yeah,
to
we'll tweet it.
look
Tweet at
into
you.
questions
We'll
and
tweet at
answer
you,
them.
and
Yeah,
yeah, not DM. Tweet at you if we
yeah.
have
Okay, tweet at
questions.
me.
And then if somebody wanted to get a hold of Fred Hutch, I think their Twitch is just at Fred and then 2 underscores Hutch. And then their Twitter is just at Fred Hutch. Is that correct?
I think that's
There we go.
correct, but
Yeah.
yeah,
Cool. That is correct. Cool. Well, thank you, Kelsey. Thank you so much. And I just wanted to say for my community, thank you guys so much for attending and asking questions and helping to raise over $200 for leukemia research. I am so thankful for all of you. I'm thankful for Kelsey, and we've got Kay Hansen too at, I'm sure it's like fredhutch.com, I think. I will, if you would like to email Jackie or Kirsten, you can email them at khansen2 or at jmontstr at fredhutch.com. Yeah, it's you. Yeah, Kirsten, it's just links. Don't worry about it. Sushi's got you. Sushi's got you. Um, I will send it to the UK MedDibs. I sure will. Awesome. Thank you so very much, Kelsey. I really appreciate
yeah.
your
Thank you for
time.
having me.
Thank you for asking questions or for answering our questions. I know, like,
Yeah,
there've been some people in chat and in Discord who have really wanted to get good answers from a trusted scientific resource who has the experience in
yeah,
this. So
yeah. I mean, I hope I was able to answer everything. I know, there are
more
always going to
than
be things you don't know. So
yeah, there's that, and like
yeah,
I said before, that is the mark of a good scientist. The mark of a
mhm,
good scientist is knowing where your boundaries are and being able to say,
yeah,
I don't know, but I can find out, or this is who you talk
yeah.
to.
Exactly,
So,
exactly. Yeah,
well, thank you, everybody at Fred
yeah,
Hutch. So we will hopefully see you another time, Kelsey.
yeah. Thanks for having me on.
Thank you.